I'm kind of embarrassed at this first slide. I usually have a picture of a really cute little piglet or something, and this is not meant to be a corporate pitch. This figure actually is really useful into how I've managed to organize and compartmentalize some of the different applications that livestock really engender. First off, it has to do with fundability, and frankly, I'll get in a little bit more detail. Mouse models, although great for basic science, and actually I got my PhD in a department that was all about model organisms. It was Drosophila and C. elegans, and I understand the power of models for figuring out basic mechanisms. But frankly, the mouse model has not been such a good predictor of the efficacy of therapies when it comes to time to test drugs or deploy them. And so pigs always, I think it never surprises anybody when I say pigs are more like people than mice, okay? And there's even colloquialisms in our language and culture about pigs. It's pretty striking. They're monogastrics like us. They're the same size as us. And for lots of other reasons, they're really likely to be a superior model for the development of therapeutics. That actually is the revenue driver for the company. And does this show up? No, it doesn't. I guess the green one does. But just that's the focus of Surrogen. We're developing swine models of human disease and providing those as platforms for developing therapeutics by academics, by the drug companies, by the device companies. Along with that, really, early on in the life of the company is Executive, which is more focused on the agricultural applications, where, frankly, I didn't grow up on a farm. I grew up on the side of a mountain. But it was a good friend of mine who's now joined the company who grew up on a Red Angus farm, and actually a Red Angus genetics company that his parents owned in Minnesota. And I got to understand the concepts about the genetic value of an animal or specific alleles in animals and the impact that had on production. And as I'll get into, there's all kinds of opportunities in trying to improve the health and welfare of animals as well as protein production. And I think as some other people were raising, actually we can do this in a way that sustains diversity, enhances diversity in some cases. So we'll talk a bit about that. And then I never thought it would be part of the company, but frankly, by the way, this is the same platform across the board, right? These are just different applications. This is just editing and deploying it for different uses. And Regenivita, frankly, I had some experience in the xenotransplantation field and walked away from that field feeling really defeated, because the more antigens you get rid of, the more there are, and just so many hurdles that I never really saw much progress in the last 15 years of significant worth. But some crazy experiments some colleagues of mine did have opened my eyes to the opportunity to use pigs as an in vivo system for the development of therapeutics. And I'll talk a bit about that. Well, so the star of the shows, of course, are the nucleases, and this is the point when I'll just say I'm an agnostic about platforms. I've had the good fortune to read some manuscripts about other platforms that are coming, and we're tool makers and users, and I think we're all going to be very surprised where this all is at in 10 years, and I don't think any of the tools are going to look like they look right now. But it's certainly an exciting time, and the democratization of these tools has been stunning to watch. So we use the technologies, the same way everybody else does. It's either the Talons or the Cas9s, and we use that to either introduce a double-strand break or a nick, and then on the basis of that are targeting the inactivation of a gene by having that nick or break in the context of an open reading frame. Or more exciting and more interesting to us is the speed with which we were able to develop the methods for what we've termed non-meiotic allele introgression, which sounds really clumsy and nobody else in the field uses it, but I can assure you it's a term that I had to develop to initiate the conversation with my quantitative geneticist friends in the livestock genetics field, in which now is opened that door to discussion. And so we'll talk a bit about that. So how do we deploy the technology in livestock? It's pretty much the same, whether it's pigs, cattle, sheep, or goats. We either can modify cells in culture. We can then clone, just like Dolly the sheep, the somatic cell nuclear transfer or chromatin transfer. It's not very efficient. Particularly in cattle, it's about 10% of the embryos that get put into recipients result in the birth of an animal, and they're not always all that healthy. There's reprogramming is not perfect. Actually, it turns out to be that's why it's so important that we've been able to resurrect the other methods for doing accelerated genetics. That is, we started out the field about the same time the first transgenic plant was made. Transgenic animals were made by injecting either DNA or then enzymes into the embryos instead directly, modifying embryos. And that turns out now using these enzymatic methods to be a lot more efficient. Went from being about 1% of treated embryos giving an outcome to now, as I'll show, it's on the order of 75% or even higher injected embryos having a modification. And then I won't talk about it much, but actually one of the methods that I think I described it in one of the companion papers, the concept about a horizontal distribution of genetics once one can develop them. And in fact, these technologies really open the door to that. Opening up germ lines for the transplantation of germ cells instead of using sexual reproduction. So I won't talk about that, but that example's given, and so you can appreciate can rapidly increase the frequency of those animals in a population. Well, so the papers, that I pointed to really, for the most part, this data corresponds to that, and these were early days. I guess it was probably a year and a half before we published this paper that we had these results on really high efficiency modification of cultured primary cells. So it's always primary cells that are cloned to clone animals. And that has represented a challenge, a difficulty, because the cells, by the time you select them out of non-modified background, will tend to senesce. And it's really dramatically decreased the efficiency of cloning. This was fast, it was efficient, and as we describe in the paper, the biallelic frequencies were far higher than we anticipated. So really one of the limitations in the genetic improvement of animals for any application has to do with the generation interval. And so being able to get the homozygosity quickly or efficiently put together different genetic backgrounds at with heterozygous changes represents a real opportunity for fast multiplication. So I guess the part that wasn't in the paper was this block that's in green here, and this is the day that we started using actually RNA encoding talons in our cells and saw a dramatic increase in efficiency, of course. And it's not in this paper, I guess it's in the next one where we start doing meiotic introgression or gene conversion. We've even been deploying CRISPRs. One of the first easy ways to do it, as we mentioned, is that you can modify cells, which is the results I just showed you, or there's modifying livestock embryos. And in fact, genetics has moved around amongst the dairy industry by way of embryos. So it's semen is sold and distributed, but so are embryos. Specific genetics are put together, and then those embryos are distributed and put into surrogates by it's in vitro fertilization and then embryo transfer. So we played around with some embryos that we developed in vivo, or should be in vitro, and early days had some rather low efficiencies modifications, but with subsequent generations of the talons that we're using, as I said, got out in the order of 75% of the embryos had at least one modification. and a significant portion had two modifications. It was also pretty high for pigs. Pig embryos are just a little bit wimpier, but showed the introgression of small indels, a third of which were biallelically modified. We followed on with the creation with the animals out of these edited cells. And I think so it represents some of the first gene-edited animals. Oops. But again, as I mentioned, it was really the fun part was when we started trying to get homologous recombination to work in the lab. And I guess you've seen these a number of times, these surveyor-type assays, where we're detecting non-homologous end joining events. In this case, we also provided, along with our talons, an oligonucleotide corresponding to the introduction of four nucleotides creating a BAMH1 site that will have interrupted the open reading frame. And to our surprise, we saw this around 40% of the cells in culture had a homologous recombination event, and which is essentially about the same as it was just observing cleavage at all as with non-homologous end joining. That seemed unbelievable, given we were having frequencies, of one on the order of 1 in 10 billion using prior technologies. So got pretty exciting, and we ran a whole series of additional loci that demonstrated the same thing, and actually what some of the early data coming out of various labs, including the one at Sangamo, Phil Gregory's group, had shown that talons worked better at cells incubated at a little bit lower temperature. And we were able to show at numerous loci that indeed the RNA, as I showed in the bottom of the one table, works better than DNA encoded talons in these cells, and that elevated temperature resulted in an increase in the frequency of either non-homologous end joining events or homologous recombination. And so again, there's a table that really describes a whole series of different homologous recombination events corresponding to even a single nucleotide change. We've introgressed some specific mutations, naturally occurring mutations that are found in livestock species. In some cases within species introgressions, and in some cases across species introgressions. I'll share some specific examples of the kind of traits that we're working on here. I'm going to summarize all that table, though, just to point out that we were really getting quite high efficiencies of gene breakage, and I guess this is modified versus homozygote. So the blue bars represent the frequency of detecting at least one introgression event, one homologous recombination event. The red is actually isolating homozygotes. Of course, what we noticed right off the bat was that the frequency of recovering alleles with insertions or deletions was higher than when recovering single nucleotide polymorphisms. And of course, it actually makes sense. The tal effector nuclease has a spacing requirement, and when you're inserting or removing nucleotides at the cut site, you now decrease the efficiency of the enzyme at re-recognizing and recutting the allele. We actually, and with a single nucleotide polymorphism, just not so lucky. We've had to develop additional methods to increase the frequency with which we get either single nucleotide introgression as a heterozygote or a homozygote. And it has to do with the fact that gene editing is a dynamic process, and we see DNA being cut and either going down one path or the other, and we've observed plenty of alleles that correspond to more than one event happening at the same locus. And so we've really tried to develop methods for skewing towards the homologous recombination events, and really did some deep sequencing analysis on modified cells to sort of differentiate between clones that had different kinds of outcomes, whether heterozygous, homozygous, non-homologous end joining on one allele and an introgression on the other. In some cases observed even more than one homologous recombination event followed by a non-homologous end joining event. and really categorize those with regards to the efficiency, the frequency that we observed it. And really we're, particularly as we'll get to the agricultural applications, I'll point out to you that we really just want the natural allele, the allele that we've already been eating. So we need to understand the frequency with which we're getting those specific changes. And we end up with complex alleles here with, the intended SNP and an unintended SNP. We've got intended SNPs with indels and just those few examples in the case of single nucleotide polymorphisms. All by themselves. So as you can see, we end up having a table of cellular clones with different allelic configurations that we have to then go through and select on the basis of what it is we want. Because of course the next step gets to be expensive, which is turning them into animals. So we've done some other things to really change the ratio and try to drive towards homologous recombination, and that can include something as simple as having the polymorphism you're trying to put in actually sit under the TAL effector domain, so that when you make the polymorphism, it destabilizes the binding site now and leaves the allele untouched from subsequent recutting. It definitely makes a difference in recalcitrant sites responded well to that. We did play around a little bit with CRISPR. I have to say that so far we're not very good at it. We get lots of great cutting. It's a very effective enzyme, but so far in our hands, in these cells, these primary livestock cells, it's the efficiency of homologous recombination that in our hands doesn't approach what we've been seeing with the talons. So we actually have another system that we're trying that's got an additional nuclear localization signal on the CRISPR, and we're hearing some good results from some of our collaborators in zebrafish. So we're looking forward to giving those new versions a try. But then there's some examples like this where, we actually go through and identify clones that have made a change we want. In this case, this is something called calipige, and this is a trait. If in French, it means beautiful buttocks, and it makes sheep have a really big rear end, and it's so more meat per animal, and the sheep it showed up in a spontaneous mutation was called solid gold, so you can imagine. But we had a really difficult time getting just the allelic change we want at this locus. In particular, we would get the allele, but we always got this compound thing going on next to it, some non-homologous end joining event next to it, and it really was confounding. So we did a couple of things. One, we went ahead and developed now different the recognition domains such that it was already a little bit destabilized. You can see we've introduced now down here either some slop either in one, another, or both two locations on this particular, on the right monomer recognition. And we end up actually getting much better. And you can see, by the way, they seem to measure, even with those changes, they seem to measure, on a population level, they seem to be functioning about the same. That's just detecting non-homologous end joining. But this change, I think this was indeed going to this, I think it was C, resulted us getting all of the clones actually being right. So it's just essentially on the edge of working. It works globally pretty well, but it's destabilized enough once the change happens that we're able to get with fairly high efficiency, and fairly high, 3 clones out of 15. So at least we're getting clones. We had another locus, a DGAT locus, which is a milk production trait. Volume of milk and protein content in milk, and the same sort of thing. It was only after we made destabilized versions that we were able to get cellular clones that just had the single change we wanted and nothing else.
So can I ask a question?
Yes.
Is there a reason not to put additional changes in the donor to prevent the recutting?
Well, I think they're political, not scientific reasons. when we're dealing specifically with, well, as we get to the agricultural part, I mean, this whole discussion went to, and I think Dan took it to, when you're trying to introduce novel traits, novel alleles that there's no evidence for us having consumed before, that's one bucket of regulation. And when it comes to food, though, our focus is on alleles that we already know exist and have a specific outcome that we've been eating for 1,000 years. Okay, and so we actually are trying to be puritanical about that. And I think clearly being able to do that has implications for lots of other things as well when you get to human medicine, and that precision and nothing else is important. Even models for the development of gene therapies, right? If the DNA itself is a target for a gene therapy, then you want the genetic constitution to be identical to what's in the patient, not just the protein that it would encode. So, well, and we've done our share of playing around multiplexing. We've put together multiple tau effector nucleases in cells, and you largely develop these models that are compromised for immunity to RAG2 and IL-2R gamma. We've put together mixtures on that basis, and frankly, just testing all the talons we had that work well together. Fairly high efficiency. We haven't pulled out the robots, so to speak, and nobody's got carpal tunnel syndrome, and we can always find the allele we want. So in this case, it's an APC P53 composite where homozygous loss of APC results in death. So we can't have that. We need a heterozygote, but we want an homozygous inactivated P53 allele, and so really only in a matter of 45 clones, I have a cellular clone that then I can go turn into a pig that has precisely the allele I want. And we've gone so far as to do 5 genes, and so this would be 10 loci, right? 5 genes, 10 alleles, where we've been trying to add it up to 5 loci at the same time, and had some success. Again, this was all in a 96 well played, I guess, in each of these examples. 4 pretty reproducibly, and again, this isn't just gene breakage, this is introgression, and seem to be doing with fairly high efficiency. Clearly, we're pretty excited about the ability of CRISPR to take this even to the next step, and as we'll talk about in the ag section, I'm particularly interested in using these editing tools as genetic tools to combine with quantitative genetics. But we did look at those cells, about 50 spreads, and didn't find any gross rearrangements. We thought with all these active enzymes that we'd end up seeing translocations or inversions, and we didn't see any of that. And so now I guess I'll talk a little bit about each of the subsidiaries and some of the products that we're focused on. I think Surrogen's it's an easy one for people to understand, and we do use animals in research, and so there's bacon for insurance. And so again, the argument being that pigs are more like people than mice are, and with the conserved physiologies, and there frankly wouldn't be devices if it hadn't been for pigs as a resource for the development of those devices and coatings of those devices and surgical techniques. And in fact, Galen was forbidden from dissecting corpses on penalty of death, but learned what he knew about the cardiovascular system from pigs. So it goes back a ways. And so it's a straightforward model that we've identified genetics that are responsible for disease in patients, that we can replicate that allele precisely. So now not your typical old-fashioned knock-ins or knockouts. This is precisely the allele that corresponds to human disease. And then they use that as an efficacy and safety testing platform to accelerate the successful traverse and development of therapeutics. The average cost per drug to go through the whole pipeline is $2.5 billion and has a 91% failure rate. So we are arguing that, use my pigs, fail early, fail often, pass, and to buy the pigs. And so So we, of course, we were a young company, and frankly, I'm an academic. I was a professor at the University of Minnesota and starting a business. I went for the sure thing, which is heart disease and diabetes, since it affects so many people, and set out to develop some models that we thought would have relevance. One of them, this is done in collaboration with the Mayo Clinic, was focused on this familial dilated cardiomyopathy. Now these patients show up as heterozygotes, usually men in their 40s. They have a heart attack, and sequencing of genomes we found to have dilated cardiomyopathy, and sequencing of enough genomes revealed that there are specific mutations in this gene and other genes in different populations, okay, that cause this trait. So imagine with you, if you will, the development of a drug that's focused on dilated cardiomyopathy that is not cognizant of genetics, okay? Whereas drugs that can be developed that specifically address the genetic cause of disease, you would expect to be more efficacious or likely to deliver a result. So there are these alleles present in this gene. It's actually a splicing factor that influences the splicing of titin. And we set out to put in this specific amino acid change to reflect human disease. And able to develop talons, and we've developed an HDR allele. This wasn't the best of our talons. We get about 18%, but that is introgression of just that mutation. Actually, it's a few mutations here. There's the causative one. In this case, we don't care, right? If we've got extra alleles, because it's not a gene therapy model, it's not a food, and we do stabilize that introgression with some adjacent polymorphisms. Indeed, we end up getting those cellular clones, and we clone them and turn them into animals. We made both homozygous versions and heterozygous versions, and made the animals. I even got some compound heterozygotes that had alleles we didn't intend, but were nonetheless inactivating alleles, and made animals. And well, the bad news was, it works. The homozygous animals born with dilated cardiomyopathy, even as newborns, actually are a neonatal model of heart disease, and actually was precisely the thing the clinicians that we were working with were after. They're actually doing stem cell transplantation studies into these piglets. We're delivering pregnant sows to them, and they're trying to save the babies with the stem cell transplantation. But of course, the people that showed up, and the way they found this mutation was adults showing up, and we'll get to that in a minute. But yeah, so the homozygotes were basically either stillborn or died within a week mostly. We have some compound hets that made it through, and some just straight heterozygotes that make it through just fine and represent our breeding herd. But just as an analysis of the outcome of this, we've looked at the splicing of some of the genes that are involved or known to be targets of this splicing factor, and showed that they are not appropriately spliced. So we're expected to get dilated cardiomyopathy, and indeed characterization of I don't know what I'm looking at here with these various tissue stains, but I'm told by our histologists that this represents dilated cardiomyopathy in these baby pigs. But what about those live animals? And we're pretty excited that actually we're getting out. I think this must be six months of age. We're actually in the heterozygous animals observing dilated cardiomyopathy coming on now, which is interesting, right? We've never had a way to watch the progression of dilated cardiomyopathy from absence to having it. And these animals are going to provide that opportunity. They're already showing troponin and an elevated expression of this brain natriuretic peptide in the blood. Well, so another area that again in the area of cardiovascular disease, we're looking at is atherosclerosis and atherosclerosis confounding factors, which is type 2 diabetes. And for this, we actually found a special pig. It's a pig that got dropped off by the Spanish on an island off of Georgia about 400 years ago, whose diet for 300 years was feast or famine on acorns. And because of that, this animal puts on weight as soon as you give it any calories. And it gets all 7 hallmarks of type 2 diabetes, including atherosclerosis. And it gets the atherosclerosis in a way that's diffuse, like us. Unlike, you can make other pigs get atherosclerosis by feeding them cheeseburgers for 2 years, but it's very focal. It's not like us. And so these pigs actually turn out to be pretty special. You do have to feed them a high caloric diet for a year to get them to get the atherosclerosis. So that's one of the things we took on, is by a couple different methods. I've interfered with the cholesterol recycling into the liver. And in this case, the first one, this is not an edit, this is a transgenic. We put in a gain of function mutation of PCSK9 by Sleeping Beauty transposition. We made an edit on the other part of this pathway, which is essentially PCSK9 controls the recycling of the LDL receptor, and expression of that hyperactive version that causes familial hypercholesterolemia hyper-recycles that receptor, so it's never there. And so the liver can't take up cholesterol from the blood. So the other way we've done this, of course, is just to knock out the LDL receptor. We'll get to that. And just to show you, we put a lot of thought into this. Nobody'd ever made a transgenic pig that expressed this just in the liver and this gain of function mutation. And we were happy we made some regular pigs and some Osaba pigs, and early results looked good. You can see that these animals had elevated cholesterol, and these are clinically relevant levels that these animals have on, this is just regular pig food, no high cholesterol diet. And it just goes on as we've been characterizing these animals. They have early onset and progressive atherosclerosis. But some early endpoints, it was up to 90% occlusion of the vasculature, and by 14 months of age. And now we've just been completing longitudinal studies that really characterize in much greater detail. I'll skip over a lot of this, but just to show that we really are able to now deploy. This is called a method called OCT, or there's an intravascular imaging system where we can actually go into living animals and look at atheroma formation. You can see there's some fibrosis here. This is early on, and even later you can see this, severe atheroma formation and the onset of fatty streaks, and there's actually lesions that look just like patients' atherosclerosis, and it goes on, and I think where we at here, this don't see what month this is. This must be our 7 or 8 months. You can see just everywhere we look, it's this atherosclerosis in accelerated manner. And he's just moving through different parts of the heart, and this is never a model like this hasn't existed, where we frankly are finding atherosclerosis in every place we look, including, so this at 9 months of age, not just different parts of the heart, where we've got foamy cells underneath and necrotic centers. We actually are seeing lesions in the distal coronary arteries as well, and there's a big foamy center underneath that forming atheroma. But the home run is frankly this one. The device industry's been looking for a large animal model of atherosclerosis in the femoral arteries. So they've moved out of the heart. They've made a lot of development there, or a lot of progress with those kinds of devices. Now they're moving into the periphery, cerebrovasculature, and in this case, looking at the femoral artery. And we're happy to say these little piggies get the same kind of disease we do, and we're hopeful this will be a product we can do well with in the market. I won't go through it, other than just to tell you that we've also, as I said, knocked out the LDL receptor, and it's not exactly the same outcome. So it's an accelerated atherosclerosis model. It's actually more severe early, and so we're interested in now making the compound heterozygotes between these animals, and really to find out what can we dial up for customers in terms of severity and timing and location of the lesions. And then this is actually hot off the press stuff. Actually the most frequent congenital killer of people is polycystic kidney disease. It's because it's never diagnosed. It's not till later in life, and the outcome is kidney failure, ultimately. There is no animal model, none. And so it's been difficult to develop therapies. So we've copied the mutations, which are well known in patients. By the way, this is what happens ultimately to the worst of these patients. By the time they show up, and that's a kidney, right? The normal kidney's a lot smaller than that. But, yeah, it's very painful, obviously, for these patients to go through this. So we actually came across there was a report in a veterinary journal about pigs that showed up with polycystic kidney disease by appearance, and they went in and took a look, and they seemed confident in this paper that was a naturally occurring mutation that caused polycystic kidney disease. So we went ahead and working again with a colleague down at the Mayo Clinic who had identified one of the alleles, several alleles for polycystic kidney disease. He's been making mice and not getting much outcome, but there's a mutation and there's a stop gain, and there's a couple of what are proposed to be hypomorphic alleles that have been identified in patients as well. And he made some predictions about the predicted severity of polycystic kidney disease given different allelic configurations. So we put those together in pig cells, and we're in the process of building that population. But again, it's like the good news, bad news. We were actually able to do a little competitive homologous recombination with the different oligos for the different sites, and we're able to get homozygotes of each versions and heterozygotes, and even some that are on one chromosome versus in trans to each other. And so it's going to be a fun genetics experiment. The sequence of those polymorphisms having been characterized by, but then our first try so 4 embryo transfers, that's into 4 recipient sows, and we got pregnant, but then nothing. And so we made some more and went in just a little bit earlier, and the good news, bad news, they were dying just towards the end of gestation, and they've got these, of course, giant kidneys that are a normal kidney. Where is it? Didn't we have a little picture? What is it? Oh yeah, that's a normal kidney size, just as a frame of reference. We're interested now. We've got this is now represents a justification for us to go ahead and put more resources in developing these lines and figuring out how we can provide them to the drug industry to finally have a target, a model to develop therapeutics to run and test their libraries against for a way to treat this disease. And of course, we're going to be tricky. It's going to be heterozygotes, and even some of that germ cell transplantation stuff I was talking about. Well, off just a little bit into Excelogen. Again, the idea being that there are going to be models of human disease that are going to be best replicated in a large animal for the development of therapeutics. We tend to try not to make models that are already, well replicated or exemplified in other model organisms. But there are these special opportunities. But really what got me psyched to work with livestock really was food. It was that exposure in grad school. It was the Christmas vacation with me and my Birkenstocks in grad school getting taken down to a Red Angus farm and slopping around in cow pies and sticking my arms up the vaginal canal and wrapping a chain around a calf and pulling a calf out. And that calf had double muscling, and it was a crystallization event for me, honestly. It was, and so food just became such a compelling subject to me, and I'm a geneticist. Like I said, I got my PhD working on zebrafish or criminy, but the livestock side just really appealed to me. Well, and of course, you've heard all this already before in Dan's presentation, and one of the compelling things is, well, we're going to have 10 billion people by 2050, and you look around and what's going to happen in politics when there's that kind of food instability. The Arab Spring was actually precipitated by an increase in the price of wheat. to mothers who couldn't afford to make bread. So this is what happens when there's not sufficient food, and so we have an interest in doing our part to embrace technology, and we're not saints either. I mean, frankly, there's a huge business opportunity as well to provide food to people who want it, and there's a dramatic projected growth in terms of the demand of meat and milk proteins and animal proteins. And so, is there a way we can produce that in a way that's consistent with principles of sustainability, of animal welfare, and cognizant of the price that people can pay? So those are some of the problems we've taken on, and I really did get very interested in genetics, and there's a long history of statistical genetics in agriculture and in livestock in particular, where taking information about pedigrees and the phenotypes of animals, designing breeding programs using statistical methodologies to produce genetics that have increased merit. And we've actually done really well with this, and in part it was facilitated by the development of artificial insemination. Artificial insemination was developed in cattle first before it was in human medicine. Okay, and what AI effectively did was to shrink geography, because now suddenly that bull could impact genetics all over the place, because you could send that little straw of semen around the world. So that's called shrinking geography, and it sped up the efficiency with which we could get the good bulls together with the good cows. It's not a technique that's really used for the other ag animal species. It has to do with the fact that those semen from those other species is pretty wimpy. For whatever reason, cattle semen is pretty tough. So. And then just to show you that selection works, it's just showing here the number of dairy cows in the thousands, and then this is the total milk production. So starting back in 1960, and of course you can see the milk production just going through the roof here because of the implementation back in the 60s of these quantitative genetic methods to say this animal has a better estimated breeding value than that animal. And they're selecting on amount of milk, they're selecting on protein content, they're selecting on the shape of the animals, okay? How many good udders versus not, and just a whole battery of traits. And of course you can see that when you have better performing animals, you also use fewer animals to achieve the same nutritional outcome, same protein production. But something else happens too, and it was brought up, and that is, it's not well known, but I'll tell you, the Holstein breed is largely based on the genetic contribution of 8 bulls. The entire millions of Holstein cattle, 8 bulls, which invariably leads right to genetic mismanagement, wherein that semen gets used, taken out of the tank and used on daughters and granddaughters, and everybody's related, and hey, there's a reason you don't marry your sister. And this is the reason. So most of the time when that happens, it's at a very low frequency, like 5% or less, and once you know it's there and you have a marker, you can just eliminate those animals from your genetic improvement program. Right? They always come back to get you. There's always recessive lethals hanging around, by the way. Okay, so there's always going to be there. But this is a case where it just got too extreme. It's about 23%. Actually, the most recent numbers I saw put this closer to 28% of Jersey cattle are related to this guy called Observer Chocolate Soldier, and he has a mutation in this gene that causes the spontaneous abortion at about 90 days of pregnancy. It's some splicing factor or whatever. It's absolutely required for viability. So it costs the industry a lot of money because that animal's been taken offline and prepared. They really bulk them up with good food and get them off and getting pregnant so they can produce milk, and they come off service as soon as they abort. So what do you do with this? You have a breed that has, like I said, about 28% of the animals. We're wanting to do genetic selection and improvement of that breed going forward, and can we really afford to throw away almost 30% of the genetic potential that's already in there to get rid of that one? And the answer is no, and there's a bunch others. Okay, and so one of the things we've done is to show that this is a simple point mutation that we can easily introgress the natural, not mutant allele to rescue a super animal when it comes along with a genomic breeding value that's off the scale, but happens to be carrying one of these recessive lethal mutations. It can actually be regenerated. So we can fix inbreeding, but we can also use this technology as a type of crossbreeding. As you can see, there are some traits that you can bring together that might be of use in one breed or another, but you'd end up bringing along traits that you didn't want to bring along, and you end up diluting purpose-bred genetics and environmentally adapted genetics, for that matter. Actually, it's been kind of a colonialist approach in the livestock industry. We developed these wonderful Holsteins or Angus cattle, and we take our semen and we go to countries around the world, and we either try to convince them that they should be using these cattle, okay, or we inseminate their locally adapted breeds, and the F1 always does okay. The F2, forget about it. This has been it's a mistake that's been repeated historically again and again and again. Right? There are genetics that are already in a location that do well there. Those animals are adapted to the disease, to the environmental conditions, to the nutritional, and to the culture. There's one of these animals you can see down here with these giant horns is one of the questions. I'll show you one of the targets for us is the genetic dehorning of cattle, and I actually was asked by a BBC reporter on the radio. He said, asking me why I wanted to take away his cultural heritage by dehorning his cattle. I don't. Those aren't the dairy animals anyway, so. But so we really have as a target a whole bunch of different traits that really the same exact traits that breeding and genetics have always had. Okay, and that is selecting animals that can, produce more protein for less input, reduced environmental impact. I mean, all these things cost money, right? This is the true carbon footprint. It's called, it's called capital. It costs money to make food. And then there's a whole series of things that really, I think, provide compelling examples of how new technologies, well, we really are in the midst of a paradigm shift in multiple levels in this field. I think it really is changing the way we think, and I hope we can change the way that other people think too, because this technology really enables ways for us to produce animals that are better for the animals too, better for the animals, better for the environment, and better for us. And I'll just give you a few examples. This is the one that struck me in the gut, which is, turns out about 5 million dairy cattle a year in the US have their horns removed. There's 3 methods. One is when they come out, they put liquid plumber on their horn buds, and it kind of digests it away, and of course it gets infected and it hurts. They can burn it off with something that looks like a soldering iron, and that's actually the way I saw it first. It was an ABC Morning News PETA video, undercover PETA person, and yeah, it turned my stomach. I mean, the guy was, burning these horn buds, and the calf is bellowing and passing out, and the guy is visibly sick himself. And okay, and I'll just tell you, then there's the third way, which is they use something that looks like bolt cutters. They do it later, bolt cutters, and then they have to reach in with a pair of pliers to pull out the veins, and blood's everywhere. And okay, so in all these cases, of course, it's lost opportunity cost because the animal is hurt and they feel it, and they're having to use their resources to repair. But the people that are doing this are good people, and no one I've talked to likes doing it. And to a person, they've asked, "Can you make this go away? Please make this go away." Well, so as it turns out, there's a natural mutation that happens in beef cattle, that makes some of them not have horns. It's called polled. It's present in really high frequency in like the red Angus, black Angus, so several breeds. It's at really low frequency in dairy breeds, 1%, 2%. That's really a contamination by beef genetics, and those animals have terrible dairy scores. And in fact, you could, just make horns go away by crossing the animals. There's a cost to that, right? Remember, these animals have been selected for different purposes. Dairy animal and a beef animal are very, very different. And we're talking thousands of years of selection. So you could just cross them, and what you'd end up with, actually we've done the numbers now, there's just not enough polled dairy animals to do this realistically. It'll never happen. There's a few animals here and there. It's just never going to go away. And you could cross them, and you'd have an animal that would be neither a beef animal or a dairy animal, and you'd watch the traits all segregate away. You've brought along 50% of the genome is now contaminating, and this is the reason industry hasn't done it, because it's economically devastating to even think about. So we showed that we could actually, I was looking for it there were basically 4 groups I knew of in the world chasing this polled genetics, and I had teamed up with all of them. It turned out it was yet a group I didn't know about in Germany that discovered the mutation, and we were pretty excited. Turns out it's 2 different mutations so 2 different occasions, mutations that cause the trait. One is really pretty complicated set of, there's an 80 kb deletion and a rearrangement and some SNPs. It's really a pretty complicated one. And then there's this little Celtic one that's present in red Angus, which corresponds to a 212 base pair duplication into a 10 base pair deletion. Okay, so a little more complicated than anything we've been doing, frankly. And it was itself a repetitive element. The mechanism is unknown. That's why it took so long to find it. It's sitting in the middle of nowhere, 2 genes off in far distance. When I've done some folding to try to predict if there's, there's a little bit of sequencing data. It may be a long coding RNA, and maybe, RNA interference. No one knows yet. I don't care. Right? I said, well, let's just find out if this really is the mutation, and does this represent a method for us to deploy dehorning. And so we went ahead and introgressed. We tried it with a couple different templates, either long oligos, or we've used plasmid DNA. We've used AAV, single-stranded DNA, as template. In all cases, we induce with a set of talons a break next to that repeated element, and by homologous recombination, we can observe between 3 and 5% of the cells in culture are actually homozygous for the introgression at that locus. So why homozygous is good is because this is actually a dominant trait. So that's to our benefit. If we can develop a few bulls of elite genetics, we could, in theory, completely eliminate dehorning from the dairy industry within a matter of, for sure, less than 5 years. Okay. So we went ahead and made some Franken bulls. We had the good fortune that this cute little fella and his brother, and there's some more brothers coming here this month. This mutation, this natural trait, has also the good fortune that not only does it result in an animal with no horns, it results in animals with double eyelashes. So it makes it extra cute, extra non-threatening, and so we're pretty excited about this. I think it truly represents a compelling example where people who traditionally find themselves on one side of an issue are going to have to embrace that times are changing, and there really are some good things we can do, the right things we can do. We can't be labeling something as a GMO. That has no meaning. Okay, it's the trait. It's the trait. What is it that you're doing? Does it make sense? And so we're excited because, of course, we can introduce this accelerated breeding method Now we showed multiple loci at the same time, or these qualitative traits. We can do it in multiple different places in an industry that, right, we don't have to reinvent the thing. We just plug right in. Livestock breeding is genetics. So that's a very satisfying thing, and we think we can do this in a cost-effective way. And again, there's lots of opportunities, and I'll show you just a few other examples. And so this is one that, frankly, is not something we're going to be spending a lot of time trying to market in the United States. There has been the selection 7 different times, 7 different breeds of cattle, all resulting in this double-muscled trait. That's the myostatin locus. It's been different mutations again, right? Farmers are just selecting animals based on, hey, I can win the state fair with that one. Okay, and 7 different times, all in myostatin, resulting in missing important residues or truncations of the protein, all causing the same outcome. The average is about, as a heterozygote, about 7% increase in retail product per animal. As a homozygote, 30%. But it's one of those double-edged swords, because I told you the way I got into livestock was because I helped pull a calf that was big, whose mama couldn't handle it because it was dystocia, we call it in livestock. The baby was too big for the hips. And the continental breeds of cattle actually all generally have small hips. So I thought very carefully, I'm concerned with animal welfare as well as producing more protein, so is there a breed that this makes better sense in? And of course, if we can control the genotype, then we can make sure that we don't have that dystocia as well. So this is, just as a proof of principle, this is a breed of cattle called Nelore. It was the breed that was brought to Brazil. They're an Indian breed. They've got you can't see it, but they've got the humps on the back. They're an Indicus breed. They're not a Bos Taurus breed. They got picked because they're resistant to disease and they can handle the heat. But they don't produce a lot of meat. But that's the #1 breed in Brazil. It's not your high-end steaks. This is your beef ingredients beef. Okay. They never had the mutation because, well, the Indians didn't eat beef. So there wasn't a selection for animals that had more meat, you see. And in any case, we just injecting embryos directly using talons against the myostatin locus, injected into some Nelore embryos, and these are actually twins. It's not a totally fair comparison because one of them has testicles and the other doesn't. Okay. But you can clearly see the difference, and this animal's hypermuscular, and we've been collecting semen from this animal now, and that provides a resource for us to distribute polled genetics if he's a homozygote, then every animal is going to make 7% more meat, no dystocia. So that's not a bad use, and particularly in let's say in Brazil, they're pretty pro-technology with regards to their regulatory. We, with partners at the Rosin Institute, took those same talons and deployed them in sheep. So there was even some allelic differences. Nonetheless, the enzymes worked, and we'll see how this guy goes. I think he ended up being, this is a spontaneous, this is a 3 nucleotide deletion. And then I don't think this is going to cause anything in this animal, but nonetheless showing that we could use the same reproductive technologies to create a sheep as well. No, those were embryo injection in that case, which is why we can't control this, right? When we do even embryo injections, we're putting in oligos, and it's about 45% of injected embryos we have introgression, not just gene breakage, and we prefer that because it's the predictable allele that we want in that case. Yeah, we haven't seen it not transmit. And cloning's 100%, right? Because the animal, but so far we do see in that we've seen some mosaicism. So there's that one animal we had 3 alleles, okay, wild type and 2 different alleles, and we make embryos or make animals, and we see we're getting transmission of all of the alleles. He's not as valuable as, say, somebody who's just got homozygous, but These are all following the expected Mendelian outcome once you look at the animals and characterize them. But this is actually something that came out recently, and I just wanted to bring this up because it in my mind, provides an example about what not to do. So pigs have been selected, and the genetics of pig production are largely constrained by what we call uterine capacity. And lots of fetal transplantation experiments have been done between different pig breeds, and if you get bigger animals inside, you get fewer animals inside. Okay, so I showed you in the case of cattle, it's one anyway, or if you do embryo transfer, you'll get two occasionally. You didn't really want that. But in the case of pigs, this group, colleague, friend of mine, who I'm gonna buy a beer for and have a conversation with, because I chose not to do this because it doesn't make sense. It doesn't make sense at all. You don't want pigs that as fetuses have more muscle. You just don't. You want the right size to use the uterine capacity. Make more litters, not more muscly piglets, because you're just going to get fewer. Anyway, and plus the visuals are disturbing. I never made a GFP pig. I never will make a GFP pig unless there's a reason. Okay, I don't want to just show I can do it to do it. And I can tell you I've done the numbers on this trait for this animal, and it doesn't make economic sense. And risk the fear, risk the ire of the public. Really we should be talking about traits that make sense. So yes.
That paper, they also had very low survival rate for the piglets. You know more detail about why that might be?
Well, so different folks use different approaches to making animals, and I would say some places are more known for brute force and some are more known for more advanced versions of technology. And I've reviewed papers where there were 4,000 cattle recipients of clones. The outcome being they got 4 that were what they wanted. So you can beat it with numbers, but to me, I want to make sure the technology is singing first before it's from a practicality perspective, and also I think, especially when I suppose you guys have talked about the human embryo editing a bit, about the ethical issues about that. At least they could have made sure the technology was working well first and then done it. I don't, anyway. So there are some other examples, and there is no pork production system in Africa, largely because of African swine fever virus. This is the Ebola of pigs, essentially. They bleed out and die within about 24, 48 hours of being exposed to the African swine fever virus. It's all over Africa, and it's actually moved into southern Europe. There are pig-like creatures, of course, in Africa, these warthogs. They get infected, but they don't bleed out and die. Which is they don't have a cytokine storm. So I really meant Ebola-like. It's that cytokine storm that results in those animals dying. These make it just fine. So there's a proposed causative allele in the REL-A gene. It's involved in inflammation pathway, I-kappa B, NF-kappa B signaling, and this allele is proposed to make it so those guys don't hyper-respond. And so we've worked with the Rosen Institute to introgress those alleles into commercial pigs. And those pigs this year will go to the Pirbright Institute where they'll be challenged with that virus. I'll say we're doing something a little different in the area of foot and mouth disease virus. Instead of sending it to the Pirbright Institute where everybody has to wear rubber and paper suits, okay? With foot and mouth disease virus resistance, we're going to the places that have foot and mouth disease virus, and we're going to make the animals in a place where we can walk down the road to challenge them. Okay, it's much more realistic and likely to result in uptake in the production system that could benefit. Well, I just want to get back to this sort of overall, this sort of global view I have about genetics and where I think this is certainly likely to go in livestock, and that has to do with the fact that A quantitative trait nucleotide has a specific effect size and it has a specific frequency in a population. I've been talking about these rare alleles that causes Mendelian traits, it's really obvious hypermuscularity or no horns, but there actually are alleles, quantitative trait alleles, as Dan was starting to allude to, that we know we can change too. And I sent an email to Dana. There's an article that came out I've been waiting for. A quantitative geneticist friend of mine we're taking some of these concepts and it actually takes very little. Changing 5 SNPs can dramatically influence the speed with which one can improve a germplasm. Like dramatically, saving tens of generations. And the magnitude of the effect is certainly larger than I ever anticipated. It certainly suggests that we'll be stepping into these low frequency alleles with intermediate effect. Of course, something that has a low effect and is at a high frequency in a population, you don't want to integress anyway. So we've gone after some of these more complex traits. In this case, it's dairy merit, which is this really, it's anywhere from, milk yield to milk composition to, time before, coming back in heat, and they're all rolled up into this one number, and you end up getting, there's 3 or 4 specific alleles in the genome that have the most of the effect. If you have all of these, that corresponds to about 30% of the genetic variance in cattle. And so we're introgressing those kind of alleles into that regional purpose-bred genetics. Okay, so take the good alleles, take just the good stuff, and give that to the folks who are trying to produce milk and protein in their specific environment. And our hope is that you can get the good results and leave some of the indigenous genetics intact. They certainly have some alleles we might need later ourselves for different environments. And, well, I'll sort of skip through this. This is my argument to the quantitative geneticists for like 5 years, and they finally started to listen because, of course, as we do this whole genome selection, as we're genetically selecting for better plants or better animals, we find out we make mistakes. There are things we didn't know. We end up down bottlenecks ourselves, and so at the very least, the technology is going to help us to prove the traits correspond to the genetics that we think we understand, but we can resolve some of those bad moves. There are good alleles that are right next to bad alleles that will never in a million years separate because of linkage disequilibrium and their proximity to each other. And so those are chances for us to make leaps and bounds in terms of animal performance. And of course, then we start getting into novel alleles where rational design opens up all kinds of concepts. But I have to say that we're focused on those, really pushing the alleles that already exist, and really all we're doing is moving the sticker. And from a regulatory perspective, that's really what we're pushing for, is that we ought to be treated in this National Academy commissioned paper, in I guess this was in Nature Biotech, really pointing out that here we can do mutation breeding and it's not regulated, and of course we've got also we've got regulated agrobacterium and transgenics but we think gene editing belongs up here with the traditional selection if they're alleles that we've been eating for 1,000 years. The safety study's done. Okay, so just like all food, we're pro post-market surveillance, and new alleles that we don't know what the consequences are, we certainly think that should be subject to some analysis. But alleles we've been eating, like pulled, for which there's such a strong ethical case to implement, and the safety of that allele is well known. It seems to us that regulation ought to come in the marketplace. It really should be post-market surveillance. That's our opinion, but of course that hits my bottom line, so it's not a surprise. That's how I feel. And I'll just say that in terms of animals, it's a little bit different. We're totally regulated by the FDA, or have been historically, but their guidelines very specifically pointed to recombinant DNA constructs. They consider the recombinant DNA constructs themselves a drug, and in the context of the animal, continues to be a drug forever. Okay? That's the article. However, we've got patients who are getting gene therapy and getting trillions of DNA molecules put into their body and then sent home to their families. And so there's really kind of a conundrum here, yes.
Is there any conflicting guidance between FDA and USDA?
No, and there is, I think, from the plant side, that it's always been that the authority with us has been with the FDA. We have in our discussions with the folks at the USDA and APHIS, they think they should be in charge. And they already have a system for monitoring the health and safety of animals and the products that comes from them. We already have that system. So we actually think they should have an authority where they don't right now. I think this statement Dan mentioned about the White House coming out with a couple of weeks ago is some good news because it's absurd, right? There's so many conflicts and we used to think DNA was magic. We spent $300 billion or something on characterizing the human genome, and we've learned a few things. And so it is time for sort of a rational look at it. And again, it's not the only fear I have of regulation is that so far, historically, it's entirely failed for animals. So there's been 2 products that have come up for consideration really at the FDA that are food animals. One was the EnviroPig. It was transgenic for the phytase gene in its salivary gland so that it had lower phosphorus emissions. Well, he was doomed from the beginning anyway because he used non-commercial genetics and the industry didn't want it, and you can just put it in the feed. It didn't actually make economic sense. And so that kind of got lost in the midst of bad economics and difficult regulation. The other one is the aqua bounty salmon, which you may know is a fish that has an extra growth hormone gene. It's got regulatory sequences from the antifreeze protein gene from ocean pout, and then the Chinook salmon growth hormone gene in the context of whatever salmon it is. And it's been under review by the FDA for about 18 years and was approved to be safe to eat and not safe for the environment, and to have agreement from the folks at the USDA and the folks at the EPA, and it disappeared. So it followed all the steps, they spent all the money, and it got lost in politics. And so that really is the challenge we all have, is that someone doesn't have to be right to really impact policy. They just have to be loud. And so we're taking a different approach. On the one hand, we're holding hands with the regulators, and they have a year that they said they're going to be taking comment and thinking about how they're going to regulate the technology, and so we're working with them to provide details to them as to how we're going to be distributing the genetics that we've created. But I fully intend to really take this to the market. And we'll just have to see what happens. And it really we're following a paradigm that, well, I don't know what that little strike under the grass exemptions is, but anyway, it really is. This is already a regulatory mechanism that's in place, and it was those things that you've been eating since 1958 that have been in the food chain. We have to assume are safe. And so it isn't a perfect fit, but we've been eating pole genetics for a long time. Usually in our burgers, not our milk. Nonetheless, it's been around for 1,000 years. And so I think this just presents just such a compelling case, right, of naturalness, of history of safety and of ethical compulsion that I'm happy I'll be submitting it by this mechanism, and if the other doesn't work, then I look forward to having a public discussion about this one. I think this one hits me, at least it hits me right here, and we'll just have to see. What I don't think any of us can afford is for this to go dark too, right? We really have to hold our politicians accountable and make sure there really is rationality. The scientists should be helping define, what's safe and reasonable. Of course, in engagement with our society and culture, but we can't afford to sit around with our mouths shut on such compelling things like this. So
the counter-argument will be so compelling that once you trailblaze with this, that other people will have an easier way to follow your path.
yeah,
You will face the challenge.
that's right.
It's so compelling that people will see, oh, when we have this, that you should succeed.
Yes, yeah. Yeah, the danger's not this product, it's all the other products that'll fall in behind it. And yep,
And that's
yep,
that's
well, they can use the argument, but it's just what it is. It's let's get real, people. It's not a GMO, it's a polled. It's what's the trait, what's the DNA, what's the change. Everything is different. You can't be thinking about things at such a global. And I tell you, if we've been eating it for 1,000 years, then by golly, it should be treated a little bit differently. It's really kind of the far end of the continuum. If we can't get that to go, we're all screwed. So this has to do again with this concept of, it's really compelled. I thought I had a table in here. Essentially, that the waiting list for organ transplantation continues to grow. And people die on the waiting list for just even the solid organs that we're all familiar with. So whether it be kidneys or liver transplantation, diabetes, of course, playing a role in both of those. Heart transplantation. I actually, when I was an undergrad, I decided not to become a doctor because I sat in on a 12-hour surgery and heart-lung transplant of a cystic fibrosis patient, and it was just a little girl. What an amazing thing to watch, and I just didn't think I could be on my feet that long. But people die waiting for organs, and so this really comes from this concept that is there a way that we can use IPS with all the developments in IPS, and I know you guys were having some discussion about this earlier. Is there something that the tools that we've developed in livestock can play a role? It also relates to my spending a lot of time working to develop iPS cells with pigs and ultimately failing abysmally. We could never get the stable line and propagation was difficult. So then I had to think about human cells instead. So the idea here is that we can differentiate iPS cells by culture techniques to turn into specific lineages and even build specific proto-organs in culture and tissues, and a lot of progress has been made, but there's also been a lot of failure in things that look right that they're just not quite right, and we don't know why. And so I'm more of a black box approach. We figured, a piglet makes organs right every time, all of them. And is there a way that we can use that as a venue? And it really got informed by some work done by some colleagues of mine that they were really suggesting that it might be that culturing essentially in vivo of an embryo might be the right environment for the development differentiation of IPS into various lineages. In the in vivo environment, and that we're having now some understanding about the genes that are involved with the development of organs and the establishment of axes and the establishment of the body plan and differentiation down the 3 tissue lineages, down mesodermal lineages, and really that deeper understanding that's come out of studying mice. Can we use that as a way to facilitate the development of therapeutics? I'll sort of skip through, and so we were pretty excited to see some of the work that's been coming out the last 5-10 years on building chimeric animals, using chimeric animals, again, putting these stem cells in the context of a developmental environment to where they can achieve their potential for specific lineages. And what's been seen is, especially in mice, showing that you can, by inactivating genes, you can open up a niche that stem cells, again, whether it's been shown both within species or now across species, rat mouse work, okay, show that a stem cell with pluripotency can both become the tissue you want, but also when you've opened up a lineage, when you've opened up a whole developmental program, they rush in. It's akin to getting on a crowded train, right? You always find that spot that's open. Of course, there's cell signaling going on and talking, but it really is even physical. We think there's an opportunity for those cells to go someplace, and they do. And it was really this paper by some good friends of mine. So Hiro Nakauchi, who was at the University of Tokyo, he's now also at Stanford, and my friend Hiro Nagashima, who I've known for a decade. These guys did just a wonderful experiment in pigs where they showed in tissue culture, taking a pig primary cell, they could inactivate a gene, in this case PDX1, which is required for formation of both exocrine and endocrine pancreas. They could, and in this case, I did that in a cell line that I think it doesn't have a color. Yeah, so if you clone and turn that into a pig, you get pigs that die without a pancreas. Right? But they showed that they could take a transgenic pig line and isolate blastomeres out of an embryo and transplant those into the morula of that knockout animal, and that those donated cells, which have a functional PDX, actually fill the niche. That animals that get born and are viable have a pancreas, and it came from the donor. Pretty shocking. Pretty shocking the degree of completedness, to be honest. I expected just primordia or partial. I mean, some of these animals, it's just striking. They are like whole pancreas. Now, it's not all donor because there's vascular system are from the recipient. There's other cells that are from the recipient, but this really suggested, wow, this could actually work. And so we set out to start to look at this, and they got lucky. This was a gain of function transgenic cell line they made, right? They didn't have a lot of resources for editing or engineering cells at the time, so they just happened to hit a pathway or a transcription factor that could be interfered with. So we thought, what if, it's going to be more complicated than that. We're going to have to get these other tissues. We're going to have to get the vascular to be from the donor, right, if you're going to actually make this work. And there are some pathways it's going to take more than one gene, breaking more than one gene to make a proper avatar, essentially. And so we spent a lot of time then exploring this concept that, again, you break the gene in the pig cell, you make the pig embryo, and then you can put adult stem cells of various kinds into the pig with the idea that could, actually be an organ that goes back to the patient that gave the stem cell, potentially, which one would expect to not be rejected as strongly as it would if it were from a pig, per se. So we've been answering some questions. Can we put pig stem cells or should we human stem cells into pig embryos? Do they propagate? Can we effectively knock out different tissues and use pigs as at least a model or a platform for the development of this technology? And just a few points that indeed we can put cells into the pig embryo. These are human umbilical cord stem cells. We put about 10 in. They go through a single doubling, essentially, and this is after 7 days of culture in vitro. They start off all over the place, but ultimately, by day 8, day 9, they really have focused in the inner cell mass, not in the trophectoderm. And this is staining here. This is human nuclear antigen. You can see that it's in the inner cell mass, and we have experiments showing the contribution of those cells to specific tissues in pigs, and I'll share some of those. So even just these are now with parthenotes. These animals could never develop into a critter. They're parthenogenically created embryos. We put think these were the umbilical cord stem cells in this case, and we actually, these are pig fetuses that have got human stem cells that are incorporated in this case in the liver, and now standing for human nuclear antigen. Indeed, we find just like in mice, and there's had been some experiments putting human adult stem cells into mouse and rat embryos, and they do populate. They go places. They go all over the place. But what about if you're if you're compromising a niche? Can we focus them? Can we have them do the same sort of thing that my colleagues showed? And so there's a few loci we went after. One is PIDX3. This is mostly because it's a visual cue you can see, and we think it has. Actually, I ended up not showing some slides for this, but essentially. There's a readout on the development of the eye that we can use as a marker for successful reconstitution of that tissue. It's a little bit controversial, and I hope we can have some discussion about it. That gene is also required for formation of dopaminergic neurons, and a cell type that has unfortunately in vitro not proven to yet go all the way. And so it's we have a member of the group who is really focused on coming up with therapies for Parkinson's. So we've also focused on some other genes in various tissues. I think in this case, I want to show this PIDX3. We actually showed that you can knock out the gene, and here's a wild type and staining for dopaminergic neurons, and when you knock it out, you don't have the lens of the eye, and there's an absence of dopaminergic neurons, and so that we're pretty interested in seeing some work from our colleagues in the neurology department on that particular model. There's another example. We've actually gotten even further with ETV2, which is an earlier mesangioblast marker. It's required for the development of the blood, as well as the endocardium, the vascular system. And so we developed talons that targeted that, and we isolate our homozygous knockouts. In this case, we had to chop the whole gene out. So it's pretty. 15% of the cells in culture were homozygous knocked out and lopped out the whole gene. And then we take and clone those, and so the picture got a little bit stretched, but here's the knockout, and you can see it doesn't have this. This is actually the allantosis, which is a highly vascularized structure that mediates gas exchange in the uterus, and ultimately, so these knockouts don't have that vascularized allantosis. They also don't have any endocardia, they don't have any blood. These are totally knocked out for those lineages. And we've gone on to do some complementation experiments. In this case, these are from GFP transgenic pig cells. We took blastomeres and did a rescue experiment, and we're showing indeed, and we're getting rescued pig fetuses that are now GFP. These are non-transgenic recipients. But wherever there's blood or vasculature, you find green. So we've accomplished that rescue with pig complementation. We've actually gone so far as to put human iPS cells in these recipients, and pretty happy to see this included in some of our recent grant proposals. We've actually got positive markers for the presence of human cells in this vascular lineages in these pig fetuses. And so these are some of the first experiments, but this complementation seems to be working. Yes.
That's fascinating. Out of curiosity, and I've never heard of this technique before, and I don't know what parthenoid is, but I assume this is an infertile pig. Is there a germline contribution? And are these things make human sperm?
No, but we could do that. No, so these are just going to be present in that animal that you make. It's not, the human cells aren't part of the germline. We do have some germline knocked out pigs, but I think there's plenty of semen out there. Yeah.
So I think we solved here it's very, very cool in the first place. Okay? But then you solve a very important question in the pluripotent stem cell field, which is the question, what is the competence of a pluripotent stem cell culture under which conditions?
Yes.
And there are people going crazy over this question,
Yes.
and you solved it. Because probably because the current literature says that only specific culture conditions will lead to contribution of human pluripotent stem cells into other species. And this problem is culture under 6i, under 2i,
Yeah. Well, I don't know what these IPS were done under. So we haven't solved it. I think what we've done is shown that there's another tool that can be used to address those questions. So we have put forward proposals where we're looking at batteries of IPS to decide which of them are better or worse at doing this complementation. Is there any difference or heterogeneity in that? But also we see this as it's a research tool that, well, you could do this in mice too, right? But there's this interesting question about there's going to be no immune response per se, no adaptive response, because this is long before the immune system exists. But there's going to be an innate immune responses. We imagine there's going to be some incompatibilities between different cell types and different differentiation programs. And actually, when there is that barrier, I think that represents an opportunity for us to understand more what some of the limitations are. So at the very least, this represents, I think, a valuable research tool. In the best of circumstances, that ETV2 pig, we could have pigs with human blood systems that feed back to surgeon as a model for the development of therapeutics that are more directed towards a humanized system. And it's just more examples of us breaking genes. In this case, we're going after the heart, and we're indeed showing that we're getting broken hearts, so to speak, and complementation. We've done it as well, gone after the skeletal markers. In mice, you have to knock out 3 or 4 genes to get ablation of skeletal muscle. And so we've simultaneously done just that. We've knocked out 3 genes homozygously in cells, and then we clone them and turn them into pig fetuses, and they have, this is a wild type and that's a knockout, and you can see we don't have, is that Desmond as a marker? Not really, don't recall, but you can see that marker, which is present, I think that's in the gut, is missing in this case from the mesodermal lineages. We're making really homozygous knockout models very efficiently, very effectively in pigs now. And here's another example where we've done a complementation with some wild type pig blastomeres. In this case, we've locked out all the musculoskeletal system, but then getting complementation and restoration of muscle. Again, providing really a rationale that this might be a system for working out details to develop a therapeutic muscle patches or stem cells. And of course, pancreas, again, just flying through these. There's where your pancreas would be. Here's our knocking out PDX1. There is no pancreas, and the staining shows that we, of course, don't have any the presence of either PDX1 or insulin in cells that are knocked out for PDX1. The liver, again, in this case it's only 1 gene, and making embryos, and they're pretty ugly and ghostly looking, but they don't have a liver. Okay, so can we do the complementation experiments now to regenerate? The liver would be a tremendous opportunity for the development of therapeutics or even toxicological reagents for testing drugs, even if we don't have the animal. We've gone on to do lungs. We knock out the lungs, and we end up, instead of getting a lung, we get just a little lung bud. So providing, we think, a scaffold, an embryonic, a developmental scaffold, as opposed to a physical scaffold, a developmental scaffold for us to develop, we think, therapeutic tissues and cells. And then that's mostly done by this group of people, and except for all we're indicated by on each of those slides, we've got about 30 investigators involved with working with us and on all these different things I've shared. And so I didn't do any of the work. I just go around and talk. So anyway, thank you. I'm happy to take any questions.